so the nervous system is composed of organs, right? So let's review just a second and go back to the levels of organization that we've been talking about since the beginning, right? So what was the smallest level of organization that we discussed when we were going through levels of organization? Okay, the cell is the basic unit of life, right? So that's the smallest level that we consider to be alive. But what makes up the parts of the cell, if you go further back than that, in the chemistry chapter? Atoms, right? So we started out with atoms, and atoms are made up of subatomic particles, and then atoms make up. Okay, so the elements are made of atoms, but what do groups of atoms make up? Molecules, right? And what are big molecules? Big molecules, the term is macromolecule. Okay, and then macromolecules make up organelles. Organelles make up cells. Cells make up tissues. Tissues make up organs, right? So when we looked at nervous tissue in the tissue chapter, the types of cells that make up nervous tissue, we have the neurons and the neuroglial cells. And then we have the nervous tissue that makes up organs of the nervous system. So what are some organs of the nervous system that you know of? You're using it right now. Your brain, right? So brain, spinal cord, right? And the special sensory organs that are part of the nervous system as well. So like in your eyes, your ears, those sensory organs are part of the nervous system. And then you also have the peripheral nerves. So the peripheral nerves are the nerves that extend from the brain and spinal cord, but they're going out to all parts of the body. All right, so the neurons are specialized to send and receive impulses. And today we're going to look at how neurons send and receive impulses. We've already talked about neurons a little bit with the neuromuscular junction, right? And we're going to come back to that because that is part of what we're going to do today when we talk about nerve to muscle interaction, but also nerve to nerve interaction. Okay, the neuroglial cells are support cells. These are cells that have a wide range of functions. They do anything from providing nourishment to the nervous tissue to protecting it. They provide a barrier between the brain and the blood. It's called the blood-brain barrier. So there's a lot of different functions there for neuroglial cells, and we'll talk about some of those specific things. Okay, the nervous system is divided up into two sections, the central nervous system and the peripheral nervous system. So the central nervous system is composed of the brain and the spinal cord, and the peripheral nervous system is composed of the nerves that are outside of the brain and the spinal cord. So you have the cranial nerves and the spinal nerves that make up the peripheral nervous system. Okay, so we're going to go through all of these within the next few chapters. This unit has 3 chapters to it. So this is our introduction chapter, and then we go into 2 more specific chapters that go into more detail. Yes?
On Wednesday, are we taking the test when we first come in, or the quiz when we first come in?
The muscle quiz? Yeah, we'll do it first thing. Okay, here you have diagrams showing the peripheral nervous system here in yellow and the central nervous system in gray. So we have the brain and the spinal cord there. And the peripheral nervous system is divided up into two divisions. We have the sensory division and the motor division. So the sensory division is collecting sensory information from sensory receptors and sending that information to the brain and spinal cord. The motor division sends the information in the other direction. So the information coming from the brain and the spinal cord goes through the nerves and out to skeletal muscles and to the smooth muscles, glands, and the cardiac muscle. So it's going in different directions here. Notice the direction of the impulse, and we'll keep talking about that through the chapter, is which way is the impulse traveling. You got to remember that sensory information is information that you're collecting from the world around you. This is things that you're seeing, you're hearing, you're feeling, you're smelling. All of those type things are coming from around you. You're picking up that information and then you send it into your central nervous system. And then sometimes you need to act on that information. Sometimes it's just something as simple as a change in light, right? If the light in the room changes, your pupils are going to change, right? So your pupils are going to dilate if the light gets dimmer. They're going to constrict if the light gets brighter. So that reaction there comes through this autonomic nervous system division of the motor division. For autonomic, I want you to remember automatic. Autonomic responses are the automatic responses that your body has to certain stimuli, right? So, for instance, as I said, your pupils dilated. Other automatic responses, when you eat food, right? The digestion process is automatic. You don't have to think about it, right? So for autonomic nervous system, remember automatic. Okay, it happens without you having to think about it. Okay, with the migraine clinical application, be sure to read through this. This is about migraines and things that are triggers for migraines. They're more common in women and are related sometimes to hormonal changes, sometimes to environmental stimuli such as foods that you eat. It could be related to an injury, something like that. So make sure that you read through the migraine section. Okay, general functions of the nervous system. So we've already talked about the difference between sensory and motor, right? And so there's this other section here that we need to mention, which is integrated function. The nervous system takes in that information and then integrates it into what you already know. You build upon it, right? So when you're a baby and you don't know anything except how to eat and sleep and cry, right? You start collecting information, and as you continue to grow and your brain develops, then you are continually integrating the information that you're collecting into your memory and into your thoughts. So integrative function takes that information and puts it in and then allows you to be able to use it, right? So it coordinates it and puts it in using to make decisions and also those subconscious things that occur. The motor function is how you act upon the information, right? So that's the information going out to your muscles and to your glands where you're able to be able to act on what you have collected. As we said, the peripheral nervous system, the motor part of it, has both the somatic and the autonomic nervous system. So somatic means body, and autonomic, again, remember, think automatic. So that's going to happen, involuntary instructions. Okay, you may remember when we looked at nervous tissue in the histology chapter. Okay, nervous tissue is made up of neurons and also the neuroglial cells. So the parts of the neuron, we have the cell body, the dendrites, and the axon. Let's take a look at that picture there. So here is a diagram of a neuron. This part is the cell body here. That's where you're going to find the nucleus. So the nucleus is in the cell body, and then the axons and the dendrites are attached to the cell body. Okay? The dendrites are carrying the impulse towards the cell body, and the axon is carrying the impulse away from the cell body. Okay? So information comes in to the cell body, and then it gets relayed out. So you think about if you're bringing in sensory information, right? You bring in sensory information, and then it needs to get sent along and go to the brain, and then in the brain you can store that information, you can process it, you can interpret it, and then you may need to send another message out to react to it. Right? Within the cell body, you also have these neurofibrils here, which are fiber filaments in the axon hillock. The axon hillock is this wide area here where the axon extends from the cell body. So you've got dendrites, you've got the axon hillock, you've got the nucleus, you have chromatophilic substance, which is basically it's rough endoplasmic reticulum that you can see. It actually just stains darker in the nucleus, and so you can see that on the slide. It's also referred to as Nissl bodies. So chromatophilic substance or Nissl bodies in the cell body of the neuron. Now, this section here on the slide is very confusing. They combine dendrite and axon. I didn't produce this. This comes from the textbook, but I think what they should have done is split this up and instead of having it all in one section. So if you use the PowerPoint, make sure that you make a note of that on here, that this is not really split up as it should be. So we'll talk about the dendrites, right? The dendrites send the message into the cell body, and then the axon carries the message away. So I already mentioned the axon hillock. Okay, collaterals are where you have branches of the axon. So it branches out there. We have the axon terminals here, which you should remember those from the neuromuscular junction, right? You have the same structure there. And then you also have the synaptic knob, which is the actual widened area of the axon terminal. So the synaptic knob of the axon terminal is this little widened, like frog finger area that we talked about. Okay, the Schwann cells. So Schwann cells are on the axon. Schwann cells are neuroglial cells that are wrapped around the axon. Okay, and the Schwann cells create what's called the myelin sheath. Okay, so the myelin sheath is the fatty area that surrounds the axon, and it helps to insulate the axon. So the myelin sheath here, we'll look at a close-up of that. The myelin sheath wraps around the axon. It contains lipoproteins. So these are lipid proteins that are insulating that axon, kind of like a cord. So when you look at a cord, like the cord here, or cord on your phone or computer, you know that you have wires inside of that cord, right? And the plastic coating on the outside keeps you from getting shocked, right? So that you don't get electrocuted. The same thing happens here with the axon. It has this coating on the outside that keeps that electrical impulse flowing through the axon instead of it just spilling out all over the place. So it helps to insulate it and to make it more efficient in that transmission of the impulse. So here we see that close-up of the Schwann cells. Here's the axon in the middle, and the axon's carrying the impulse away from the cell body, and then the neurofibrils in the axon there. We have the myelin sheath, which is this section here where it's wrapped around multiple times, and then that outer edge of the Schwann cell is called the neuralemma. Okay, and the neuralemma is the outer edge. That's where you find the nucleus of the Schwann cell, and it's just it's going to be important. We'll talk about it in a minute with regeneration of a nerve fiber. Okay, so it's this outer area of the Schwann cell. When you think about fat, lipids, right? What color is fatty lipids? Think about a steak. You cut into a piece of steak. What color is the fat in the steak? It's a yellowish white, right? So we're going to talk about white matter and gray matter, okay, when we talk about the brain and the spinal cord. And that is related to whether or not the nerves or the axons are myelinated, okay? So myelinated areas are going to show up with this yellowish white color, and we call that white matter. The unmyelinated portions are going to be gray, and that's the gray matter. Okay? So remember that when we get to the brain and the spinal cord. So this myelin sheath is the coating. It's a protective coating. It's an insulator. Okay? And in this diagram here, you have an unmyelinated axons there. It's still surrounded by the Schwann cells, so you have a group of axons, and they're surrounded, but the myelin sheath is not wrapped around the axon multiple times. It's just kind of in that area. And there's parts of the axon that are exposed. Okay, in between the Schwann cells, there's a little space called the node of Ranvier. Okay, the nodes of Ranvier are little spaces in between each Schwann cell. Right, and when we get to the impulse transmission, we'll talk about why that little area is significant. Okay. But for now, know that it's the gap between the Schwann cells, and it's called the node of Ranvier. Okay, here we've got some slides of myelinated versus unmyelinated axons. Okay, and you can see here's the myelinated axon here. Here's the myelin sheath going around it. In this unmyelinated axon, notice that there's no myelin sheath. There's still a Schwann cell there, but there's no myelin sheath. So it's just cytoplasm of the Schwann cell that wraps around the axon. But it doesn't have that fatty, insulative, and protective substance of the myelin sheath. Multiple sclerosis. So make sure that you read this clinical application on multiple sclerosis. A sclerosis is a scar. It's a scarring of nervous tissue that occurs when the immune system attacks the myelin sheath and starts to break it down, and it causes the nerves, the axons, to begin to degenerate, and muscles stop contracting and atrophy. It causes fatigue, other problems with mood, vision, and other issues that people experience. And it is an autoimmune disease. So the immune system attacks the body, not recognizing it as your own. Yes.
I have a question. Looking through your science assignment,
Uh-huh.
you didn't grade anything yet, right?
I graded exercise 19 last week, and I haven't graded 20 yet.
I'm looking at it very well, but I'll ask you on the class.
Okay. All right, the classification of neurons by structure. All right, so you have multipolar neurons, bipolar neurons, and unipolar neurons. So this one right here is multipolar, meaning that it has more than one pole or connection to the cell body. So multipolar. You have bipolar, meaning that it has two connections there to the cell body. And then you have unipolar. There's only one actual connection to the cell body right here. The processes that are carrying the impulse towards the cell body are the dendrites. The process that is carrying the impulse away from the cell body is the axon. So all of these would be going in towards the cell body and one impulse leaving. For this, you have the dendrites up here going towards the cell body and then the impulse leaving. For this, it's a little bit different because you have the dendrites at the very tip, and then this is going to be the axon here that is leading away from the dendrites, right? And it goes down the length of the axon. So these would be found in different areas within the nervous system. The multipolar neurons are your motor neurons are going to be multipolar neurons. Most of the neurons of the central nervous system are going to be multipolar. Your bipolar neurons, you're going to have sensory receptors that are bipolar neurons. For instance, in the eyes and the ears, you're going to have these bipolar neurons. And then here you also have sensory receptors that can be the unipolar neurons as well. Okay, so it's just specific areas within the nervous system where you have different configuration or structure of the neurons. So this is classification based on how it looks. Now we're going to talk about classification based on its function. Okay, so function, we can have three basic functions. We've got sensory, interneurons, and motor neurons. So sensory information is coming in to the central nervous system. This is also called afferent neurons. So this information is being collected and picked up by the receptors, and it's going towards the central nervous system. Okay, the interneurons are going to be taking in that information, the impulse, from the sensory neuron, and then they relay that over to the motor neurons. Okay, so information comes in, it gets relayed over, and then it goes out. So if we follow along here, we've got a sensory receptor in the peripheral nervous system. This could be a receptor of the skin. Remember, we talked about the tactile corpuscles in the skin, the Meissner's corpuscles in the skin, right? So these are receptors in the skin, and that information of touch can be picked up here. It gets relayed through the sensory neuron into the central nervous system to the interneuron. The interneuron is going to take the impulse and transfer it over. It can go from one interneuron to another. It may just be one interneuron, or there may not be an interneuron at all. Just depends on the location. Okay, and then the information gets relayed over to a motor neuron, and it goes out to the effector. So this is the efferent pathway here, where you have the motor or the efferent neurons that are transferring the instructions from the brain, the spinal cord, to the effectors, which are the muscles and the glands. Okay, so you have receptors and effectors. Okay, receptors are picking up information. Effectors are responding to the information. Okay, general functions of neuroglia. Okay, they provide structural support for neurons. Okay, this means that they are surrounding the neurons. These are the neuroglial cells are what we call helper cells. They're not the neurons themselves, but they help out the neuron. Okay, so the neuroglial cells are going to help to guide the growth and development of the neuron as the axon is growing through to the appropriate tissue. They help to nourish the neurons and stabilize the ion and neurotransmitter balances, and they aid in the formation of synapses. Synapses are those connections from one neuron to another. Okay, so the neuroglial cells, there's a lot of different types of neuroglial cells, and we'll look at the specific types. Okay, so this page has the information about the different types of cells. Let's look at the pictures here. So here the pictures, we can see, first of all, we have these oligodendrocytes. Oligodendrocytes make the myelin sheath in the central nervous system. Okay, I mentioned a type of cell that makes the myelin sheath in the peripheral nervous system. It starts with an S. Does anybody remember the name of that cell? The Schwann cells, right. So the Schwann cells make the myelin sheath in the peripheral nervous system. The oligodendrocytes do the same thing in the central nervous system. They look a little bit different. Notice here that they can be connected to more than one axon there, but they're doing the same thing. They're wrapped around the axon, they're insulating it, they're protecting it, and they're providing that framework for the axon. Okay, then we also have a microglial cell. A microglial cell is one that moves around through the nervous tissue and phagocytizes particles and harmful cells, bacteria, things like that might try to get into the central nervous system. So those microglial cells are specialized to get rid of debris and pathogens, okay, through phagocytosis. Remember, that is when the cell eats particles and things like that. Okay, then we have the astrocytes. Astrocytes are connected here to the capillary, and then it goes over to the neuron. Right? The astrocytes, what do you think they're doing? They're connected to the blood supply. What's the blood carrying that the neuron needs? What's in your blood that your cells need? Oxygen and nutrients, right. So the oxygen and nutrients in the blood, it needs to get over here to the neurons, right? So that it's carried through the astrocyte over to the neuron. The ependymal cells over here. So your ependymal cells are lining the cavities that are created by the ventricles inside of the brain and the central canal in the spinal cord. In those cavities, you have a fluid called cerebrospinal fluid. Okay, and cerebrospinal fluid, CSF, okay, if you see that abbreviation, cerebrospinal fluid is a fluid that circulates through the brain, through the spinal cord, on the outside of the brain and the spinal cord. It's a protective fluid that is produced from the blood plasma, okay? And it helps to regulate the flow of different ions and things of that nature, and also it helps to cushion and protect the brain and spinal cord. Within these cavities, these ependymal cells have cilia on them. Remember, we talked about cilia in the histology chapter, and tissue with cilia moves substances along, right? So remember, we looked at the trachea, and the cilia in the trachea were trapping and moving substances. Well, the cilia here in the ventricles help to move the cerebrospinal fluid and circulate that fluid through the brain and the spinal cord. All right, so the peripheral nervous system, here's those Schwann cells again. Okay, they're wrapped around the axon, creates the myelin sheath, this outer layer here called the neurilemma. Okay, remember that. It's going to be important when we talk about damage to nerves, is that neurilemma. Right, we're going to look at a slide that looks like this today. Right, and the satellite cells. So satellite cells are support clusters of neurons, or they support clusters of neuron cell bodies. The cell bodies of neurons outside of the brain and spinal cord are called ganglia. So that's what that term means. That's when you have the nerve cell body in a cluster, okay, so several of them, outside of the brain or spinal cord. Okay? It's called ganglia. And you'll look at that today as well. Okay, so neuron regeneration. We know that damage to nerves can be very detrimental, can be life-changing, can be fatal, right? But in some cases, the nerves can regenerate axons, okay? So think about if you have a cut, right, and you cut one of your one of the smaller nerve processes going to, you know, some of your muscles. That muscle is not necessarily paralyzed, right? Because the axons are coated in this myelin sheath, and the myelin sheath with the neurilemma provides a template for the axon to regrow through. So you don't always have permanent nerve damage if you have damage to a nerve. If you have a major surgery, right, they may have to cut deep within the body, or if you have a major injury, they may have to cut deep within the body. And this could cause some nerve damage, right? It may not be permanent depending on the amount of the damage, because the Schwann cells provide a guiding sheath for the axon to regrow. Now, in the central nervous system, the axons don't have the neurilemma. Remember, we talked about in the central nervous system, they have the oligodendrocytes that don't have that outer neurolemma template. And so in the central nervous system, in the brain and the spinal cord, those axons can't regrow and regenerate themselves. And so that damage is more likely to be permanent damage, such as brain damage or spinal cord damage. the mature neurons, they're not going to replicate themselves. So once neurons are produced, they stop. They're not going to keep dividing, through mitosis. Once they're produced, they're done. So you have those stem cells that are going to produce the neurons, but once it has divided and produced a neuron, that neuron is not going to continue to divide. So your amount of neurons is not going to increase as you grow and learn and develop. You've already got the number of neurons that you're going to have. It's just the connections that you make as you continue to learn and to grow. Here you can see that pathway where the injured axon would be. And so here's the site of the injury. It gets severed, and you still have that neurolemma there that allows the axon to regrow in that pathway. And then eventually it's repaired. Okay, so I'm going to stop here and we're going to look at slides for lab.